Hello everyone, this is the Chinese Mitten Crabs Aquaponics project. Today, we are going to talk about how biotechnology is applied in our daily life. The discovery of DNA structure and some other advances in science have led to a rapid development in biotechnology. It refers to the use of organisms, biological systems, or processes in producing goods or providing services. Let's look at some developments in biotechnology. Look at the two tomatoes here. They were both picked when they were nearly ripe and were then left at room temperature for 4 weeks. The tomato on the right went bad, while the tomato on the left was still in good condition. What caused such a difference? It is because of recombinant DNA technology. The tomato on the left is actually a genetically modified tomato. Scientists introduced a specific gene into a tomato plant to slow down the ripening process of its fruits. The tomatoes produced had a longer shelf life. This tomato is a genetically modified organism. It is produced by genetic engineering. Genetic engineering refers to the changing of the genetic makeup of an organism by direct manipulation of its DNA. Recombinant DNA technology is often used in genetic engineering. This is a technique in which a fragment of DNA from a donor cell or organism is isolated and then inserted into the DNA of another cell or organism. This allows scientists to introduce a new characteristic into an organism by inserting a new gene into its DNA. How does recombinant DNA technology works? It basically involved cutting and joining DNA molecules from different sources. In this technology, a DNA fragment containing a gene of interest is inserted into another DNA molecule called a vector. The vector carried the gene of interest into a host cell for expression. Plasmid, a small ring of extrachromosomal DNA, which can be found naturally in bacteria, is usually used as a vector. There are 4 major steps in recombinant DNA technology. First, obtain DNA fragments containing the gene of interest from the donor cells, which can be any nucleated cells. Second, obtain a plasmid from a bacterium. Third, cut DNA fragments and plasmids with the same restriction enzyme. This enzyme acts as the scissors and cuts open the plasmid. Finally, using a DNA ligase to join DNA fragments and plasmids together. A DNA ligase acts like glue that catalyzes the joining of DNA fragments and the plasmids. Finally, recombinant plasmids are formed. After these 4 steps, the recombinant plasmid can be introduced into host cells for expression, such as producing human insulin for people with diabetes and producing tomato plants with the characteristic of longer shelf life. Therefore, what are the applications of recombinant DNA technology? It is used in medicine, agriculture, and food production, industrial uses, and environmental protection. In the field of medicine, it has not only helped in the production of pharmaceutical products, but also gene therapy. As mentioned before, human insulin can be produced using the recombinant DNA technology. Patients with diabetes can receive the injection at a lower cost and lower risk for infections and rejection by the body's immune system, as the insulin produced by the recombinant DNA technology is pure and structurally the same as the secreted by the human body. Other than the human insulin, vaccines can also be produced by the recombinant DNA technology. Besides the production of pharmaceutical products, gene therapy is also benefited. Gene therapy is a potential cure for the diseases that are caused by a defective gene, such as sickle cell anemia. The most common therapy is to supplement the defective gene with a normal one. In this approach, a normal gene is inserted into a vector using recombinant DNA technology. The vector then carried the normal gene into a patient's cell to treat the disease. Let's move to the field of agriculture and food production. Recombinant DNA technology can increase the productivity of some crops and farm animals. Salmon can be genetically modified to grow faster. A GM salmon has a larger body size than an ordinary salmon at the same age. GM salmon usually grows twice as fast as normal salmon. Besides, an ordinary cotton plant can be attacked by pests, while a GM cotton plant does not. Pest resistance characteristics help to improve productivity. Genetically modified crops have more nutritional value than that of ordinary crops. For example, golden rice is a GM rice, in which the grains are rich in beta-carotene. It will be converted to vitamin A in our body. That helps good vision and immunity. Recombinant DNA technology is also applied in industrial use. It can enhance productivity in the production industry. Large amounts of cellulose can be produced in a relatively short time using the recombinant DNA technology for producing stone-washed genes. Last but not least, environmental protection. Using recombinant DNA technology, oil-eating bacteria can be genetically modified so that they break down a wider range of hydrocarbons in the oil into harmless substances.
This is the end of the video. Thank you for watching. Please give us a like and subscribe to our channel. Click on the tab to watch the next part of the video series.